Okay, experiment 15 is bacterial population counts, and this is one of the more confusing, conceptually confusing labs that we have. And so I'm going to try to get through this. The purpose was to show how to quantitatively analyze a sample and evaluate how gross it is, pretty much. You find out how many microbes are viable in a specific sample, pretty much. Well, not necessarily viable, I guess. Just how many microbes, different ways to find out how many microbes are in a sample. So there are three ways to do this. There's direct microscopic count, standard plate count, and turbidimetric assay. Now, direct microscopic count is where you take a sample, you take just like a little bit, it's probably about a milliliter of it, and then you put it into a little chamber, and then you have the ability to see what's ever growing in there, and then you go through each one of these little boxes or columns or something, and you count how many cells you see in this little area, and then usually you will typically multiply it times some coefficient, and you'll get some colony or some number of cells per some volume. I actually do this I work at the vet school, and we actually do this exact thing, and so it's kind of dear to me. But so it is a thing. We're not lying to you when we say that this is a direct microscopic count. We do this with parasite eggs, and it works very effective. With standard plate count, this is what we did in this lab. What it is you grow, you take a plate and then you grow bacteria on it, and then you count the number of colonies you see based off of the number of you count the number of colonies you see, and then you imply that the number of colonies present equals the amount of cells present, and that one cell yields one colony, which has some problems in it that we'll get in later, but that's the basic logic behind that. You need 30 to 300 colonies, and that's an important thing there. You can do this two ways. You can spread plate. You can just put them on top of the agar. So you take a plate that's already made, and then you just put the bacteria on top, and then you allow it to grow, and you count the number of colonies. Or you can, before the agar is solidified, you can actually combine it with the agar and allow it to grow within the agar, and then count the number of colonies. We do both of these in this lab. The last one is turbidimetric assay. This is where you put a culture into a broth tube, and then you actually measure how turbid it is relative to a blank sample, and then you do some calculations. You just put it through a spectrophotometer, and then you can actually do some calculations and find out the growth of it. And so those are three ways. There are benefits and there are advantages and disadvantages to each one of these. Direct microscopic count, it's very rapid. There's no culturing, and therefore there's no selection against certain organisms, so anything can grow. So they all grow. But however, counts are of alive and dead cells. Sometimes it requires a lot of skill. At my lab job, we sometimes the eggs we're trying to count are actually different eggs than what we want to count, and so it takes knowledge of the type of cell and what it looks like to require the count, and it also requires a hemocytometer, so you have to have that special plate. Your standard plate count, it measures only living cells, and so it needs time for which is good, and because those are the effective ones that are going to be doing things and stuff, but it needs time for incubation, and then it selects against certain organisms that can't grow. So what if you have a plate that doesn't grow a certain organism? You can't measure its because it won't produce a colony. You can't measure its like cell count. But that's just a general thing. The spread plate is relatively simple to perform, which is good, but you need a good technique, and it just corresponds to the standard plate count as well. Pour plate, it's good that there's no alcohol, there's no flames involved, so you're not killing anything that you don't want to kill by accident. But it is trickier than a spread count because sometimes melted agar can get tricky. You need melted agar as well, and then also all this stuff corresponds to both spread plate and pour plate. Turbidimetric assay, it's very quick. There's no incubation needed. You can just plop it in there and then go. But you can't use for organisms that grow in clumps because it doesn't make the suspension turbid enough. So what this relies on is dilution. So for spread and pour plate methods, you need 30 to 300 colonies on the plate, as we said here. So you need this because less than 30, the data is statistically insignificant, so it doesn't really matter anymore. But more than 30, the growth medium and the growth medium ability is called into question. So some of the nutrients might be depleted to where it can't grow anymore, and so there might be more cells on there that just don't have nutrients. But the important thing about dilution, or the important thing about these numbers, is that they're off by a factor of 10. So 30 times 10 is 300. And that's an important thing right here, is because when you take a dilution, you can take a concentration, and then if you can put it into something that divides or multiplies by 10, you can usually bullseye a number that is in between these two numbers. And so that's what's important about the dilution. So for example, if I have a liter of broth that has a concentration of 2.8 times 10 to the third colony forming units per milliliter, that's CFU per ml, colony forming units per milliliter, that number right here is not between 30 and 300. And so what you can do is you can take 1 milliliter of it. In that 1 milliliter, the concentration is still 2.8 times 10 to the third CFU per ml. And if you take that milliliter and put it in 9 milliliters of broth, then the concentration, you divide by 10, effectively, right? Because you're taking this concentration and then putting it into 10 milliliters, so you're expanding the volume. And so the concentration goes to 2.8 times 10 squared, which is 280, right? And so 280 is between 30 and 300, so you have the number that you're going for. Now, furthermore, if you take a milliliter of this and then put it into 9 more milliliters of broth, then the concentration goes to 2.8 times 10 to the first, which is 28, which is less than 30. So, so doing this dilution allows you to bullseye a number, like I said, that is between 30 and 300. So in actuality, you typically don't know the concentration of the original sample. And so what you would do is you would just take a sample, take like a milliliter of it, for example, and dilute it until you get a countable plate. So you would go through this whole process not knowing that it's 2.8 times 10 to the third, but then when you go to count this plate right here, you see that, oh, there's 280 on here, so if I multiply times 10, we have the original concentration of the original sample, and that's 2.8 times 10 to the third. And so that's the logic behind it. So you count the plate and you find that it's 280, for example, and then you multiply times 10, and then you get your 2,800 concentration. So there are different ways to dilute a sample. So like I said, you can take 1 milliliter and put it into 9 milliliters or something, and then you go from so you effectively multiply by, or you divide by a factor of 10. If you take 1 milliliter and put it into 99 milliliters or something, you divide by a factor of 100. So you see how that's just another factor of 10 right here. If you take 0.1 milliliters or something and put it into 0.9, then that's just like this one right here. It's just everything's divided by 10, but it's still the effective concentration is being divided by 10. And then the same thing right here is that if you take 0.1 milliliters, put it into 9.9 milliliters, then you divide by 100. So the last way is that if you do this 0.1 to 0.99, or 9.9, you dilute it times a factor of 10. And so one thing you can do is you can take 1 milliliter of that and then put it in an auger, and then take 0.1 milliliter of that same thing, which is 1 divided by 10, which is 0.1, and then put it in that same concentration of auger, and you'll actually get a different concentration because you use 1 milliliter here and then 0.1 milliliter here, and we end up doing that in this lab. So what we did was each bench had a sample. Bench 1 had E. coli, bench 2 had ground meat, and bench 3 had milk. Bench 4 had old milk that sat out for a little bit. And you worked in group of fours to dilute the sample and then do a pour plate method, and you count the number and find the CFU per ml of the original sample. Now you go back, like we said earlier, and you count that plate. You find a plate that is countable, that has between 30 and 300, and then you count that, and then you multiply it times this what's called a dilution coefficient, and then you find the CFU per ml of the original sample, and that allows you to see how many bacteria are growing in that thing. So in the follow-up, some of you had uncountable plates, which is, it happens in that you just have a bad experience with it. I don't really get why, because conceptually it all makes sense. But uncountable plates do happen. Just write down in your lab manual that you had an uncountable plate, and then it'll be fine for grading your follow-up. Now, the take-home message, though, is to know how to do dilution problems, because you do have it on your midterm and final, I think. So you need to know how to do this math that's involved. The lab manual goes over it in pretty good detail about how to do that math, and so make sure you check that and see how to do these problems. But the really important take-home is to recognize the logic and how microbiologists find the CFU per ml. Because it's used in a lot of studies, and it's an important thing that you should take away from this lab. So make sure you know how to do it, get the logic behind it, and then if you have any questions, go ask someone. But it is a very important skill.